Hello everyone. In this module, we will study about the two essential macronutrients, nitrogen and phosphorus. Nitrogen and phosphorus are indispensable to the plants as they are a part of nucleic acids and other biomolecules. In this module, we will discuss their role in plant growth and development as well as the mechanisms of their uptake and transport. We will also study the symptoms plants demonstrate during their deficiencies. The learning objectives of this module are an introduction to nitrogen, nitrogen cycle, nitrogen soil availability, uptake and transport, biological nitrogen fixation, Rhizobium-legume symbiosis, nitrogen deficiency, an introduction to phosphorus, phosphorus cycle, phosphorus soil availability, uptake and transport, phosphorus and role of mycorrhiza, phosphate solubilizing bacteria, and phosphorus deficiency. Nitrogen is an essential and quantitatively the most important macronutrient for plants. It exists in many interchangeable forms which are influenced by climatic and biological conditions. It is also the fourth most common element. It is found in proteins, nucleic acids, plant growth regulators, chlorophyll, and many other biomolecules. The two bioavailable forms are nitrate and ammonia. Although nitrogen is plenty in atmosphere, though it is limiting in environment for growth of plants. Nitrogen is made available to the plants after conversion into usable inorganic and organic forms through microbes driven processes. The three major nitrogen pools are atmosphere, soil and biomass. Plants convert inorganic soil nitrogen to organic nitrogen, that is amino acids and nucleic acids, etc. The organic nitrogen moves up the chain to animals as they eat plants, and nitrogen returns back to the soil in form of animal waste and decomposition after death and decay of organisms. There are four processes that participate in the cycling of nitrogen through biosphere. Nitrogen fixation, which involves conversion of free nitrogen into ammonium ion. In atmosphere, it is done through lightning. Industrial fixation of nitrogen is also carried out, and biological fixation of nitrogen is carried out by certain microbes. The second process is decay, which involves conversion of organic nitrogen into ammonium ions. The third process, nitrification, where ammonium ions are converted into nitrate, and denitrification, which converts these nitrate into free nitrogen and N2O. Nitrogen occurs as two major forms in soil, organic nitrogen and inorganic nitrogen. The organic nitrogen are the solid forms of organic nitrogen which constitute the dominant forms. They are unavailable to plants until processed into small soluble units by enzymatic activity. Most nitrogen in soil solution is of high molecular weight, while roots only possess the capacity to take up low molecular weight, like urea, amino acids, polyamines and small polypeptides. The inorganic nitrogen exists as ammonium ions, nitrate ions. Only a very small fraction of this total nitrogen is in inorganic forms. The conversion of organic forms to inorganic forms occurs via biological processes involving various soil microorganisms. The concentration of nitrate in soil solutions is variable, both spatially and temporally, which depends on soil type, the fertilizers added, as well as the microbial activity. Inorganic forms and few forms of organic nitrogen are taken up by roots and are finally transported in the xylem to the shoot or in the phloem to the growing tip. The diagram illustrates the uptake of nitrate and amino acids. Nitrate is reduced in root cortical cells via nitrite and ammonium to amino acids. In the roots of tropical legumes, ureides are also synthesized. Following the uptake into root hairs or root epidermal cells or the synthesis in the cortex, nitrogen moves towards the vascular system, which might follow the symplasmic pathway from cell to cell through plasmodesmata. In an alternative pathway, nitrogen can also move epiplasmically into the root and is then taken up into the cortical or endodermal cells. The uptake into the symplasm occurs by cells which are located on the outer side of the Casparian strip, which is shown as the grey line, that blocks inward radial movement through the epiplasm to the vascular system. Once the Casparian strip is circumvented, amino acids, ureides or nitrates are released into the vascular epiplasm by export or efflux systems for xylem loading, or they are also loaded via the epiplasmic route into the sieve element companion cell complex of the phloem. At any given point along the symplasmic transport pathway, nitrogen may leak into the root epiplasm and can later be retrieved into the root symplasm. Nitrogen in the xylem sap can be transferred from xylem to phloem as indicated by the blue arrows in the diagram. Organic nitrogen in the soil is primarily in the form of amino acids and urea, as well as in peptides and proteins. Amino acid uptake is carried out by two pools of amino acids, which are neutral and basic, that is L-arginine or L-lysine. These are transported into the symplast of the root epidermis, cortex, or the endodermis by the amino acid transporters lysine histidine transporter 1 and amino acid permease 5. Both these transporters are also expressed in the stele, suggesting a function in transport of amino acids from the epiplast into the cells. Various transporters have been known to participate in root amino acid uptake, including LHT1, amino acid permease 1, amino acid permease 5 and proline transporters 2. LHT1 is mainly involved in the uptake of acidic and neutral amino acids, whereas amino acid permease 5 is involved in the uptake of basic amino acids. The organic nitrogen acquired by roots is delivered to source leaves by a transpiration stream through the xylem, which is either metabolized or transferred to the phloem tissue for long distance transport. The amino acids and peptides are loaded into the phloem of minor veins via the symplastic and epiplastic pathways. In the symplastic loading mechanism, the organic nitrogen moved into the sieve element companion cell complex of the phloem through the plasmodesmata. Whereas in the epiplastic loading, the organic form of nitrogen is released into the epiplast, which is followed by the active uptake into the sieve element companion cell complex of the phloem by plasma membrane located transport proteins. AAP1, which has been found to be localized in the outer epidermal cells of embryos in Arabidopsis, has been found to be critical for amino acid import into seeds and also mediates seed protein levels. Plants absorb ammonium from the soil or via symbiotic exchanges with microorganisms through the root system. The preferred form in which nitrogen is taken up is dependent on the plant's adaptation to soil conditions. Mostly, the plants adapted to low pH and reducing soils, as found in mature forests or Arctic tundra, tend to take up ammonium or amino acids, whereas the plants adapted to higher pH and more aerobic soils prefer nitrate as the form of nitrogen uptake. The nitrate uptake is carried out by membrane-bound transporters, which are essential for the inter- and intracellular movement of nitrate inside the plants. The two nitrate transport systems, NRT1 and NRT2 families, have been identified to act coordinately to uptake nitrate from the soil and transport it throughout the plant. Nitrate transporters of NRT1 and NRT2 families regulate plant root cells to uptake nitrate through low affinity transport systems known as LATS and high affinity transport systems, that is HATS, depending on the external nitrate concentrations in the soil. When the external nitrate concentration is high, the LATS functions and appears to be unregulated and constitutively expressed. However, At lower concentrations, there are two HATs that operate for nitrate absorption. One, the constitutive system, and the other, the inducible system. Once the nitrate is taken up into root cells, it can move radially across several cell layers and distributed in various tissues. During nitrate shoot transport, nitrate is loaded from the steel cells into xylem vessels for long distance transport through the transpiration stream. AT&RT 1.8 and AT&RT 1.5 are involved in nitrate unloading from the root steel or from the shoot vasculature and nitrate loading into the root steel. This was illustrated in Arabidopsis thaliana. NRT 1.7 plays an important role in nitrate phloem loading in the source leaf to remobilize nitrate from older leaves to the nitrogen demanding tissues. A low affinity nitrate transporter, that is AT&RT 1.4, is constitutively and specifically expressed in the midribs and petioles of Arabidopsis leaves. The AT&RT 2.7 protein is involved in the loading of nitrate into the vacuole during seed maturation. Ammonium is the predominant source of nitrogen that is available for plant nutrition. Plants absorb ammonium from the soil or via symbiotic exchanges with the microorganisms. Ammonium uptake is mediated by the members of ammonium transporter families, which are AMT, MEP or RH, which include AMT1 and AMT2 subfamilies. AMT type ammonium transporters regulate the major entry pathways for root ammonium uptake. Transporters of AMT 1.1, AMT 1.3 and AMT 1.5 are expressed at the plasma membrane of epidermis cells and root hairs. These are responsible for the ammonium symplastic pathway. In Arabidopsis, AMT 1.2 is expressed in the endodermis and root cortex, suggesting that AMT 1.2 regulates ammonium uptake via the apoplastic transport pathway. After the ammonium is taken up by the roots, it is stored in root vacuoles and later assimilated to amino acids or transported to the aerial parts of the plant. The low affinity transporter AMT 1.2, located in the endodermis and cortex cells, has been found to play an important role in retrieving ammonium released from the cortex and also the ammonium entering the root tissue through the symplast pathways. Biological nitrogen fixation was discovered by Bijering in 1901. He found that free nitrogen is fixed by a specialized group of prokaryotes. These organisms utilize the enzyme nitrogenase to catalyze the conversion of atmospheric nitrogen to ammonia. Plants can readily assimilate this ammonia to produce nitrogenous biomolecules. These prokaryotes usually include aquatic organisms such as cyanobacteria, free-living soil bacteria like Azotobacter, bacteria that form associative relationships with plants such as Azospirillum, and Rhizobium and Bradyrhizobium that form symbiosis with legumes and other plants. Specific genes determine which Rhizobium strain will infect which legume. Even if a strain is able to infect a legume, the nodules may not be able to fix nitrogen, which are later termed as ineffective. The effective Rhizobium strains induce nitrogen fixing nodules. This effectiveness is governed by a different set of genes in the bacteria. The NOD genes direct the various stages of nodulation. The initial interaction between the host plant and free-living rhizobia involves the release of a variety of chemicals by the root cells into the soil. These chemicals encourage the growth of the bacterial population in the area, usually around the rhizosphere. The reactions between certain compounds in the bacterial cell wall and the root surface are responsible for the rhizobia recognizing their correct host plant and attaching to the root hairs. The flavonoids secreted by the root cells activate the NOD genes in the bacteria, which then induce nodule formation. The whole nodulation process is regulated by a highly complex chemical interaction between the plant and the bacteria. Once these bacteria bind to the root hair, they start excreting NOD factors, which stimulate the root hair to curl. The rhizobia then invade this curled root and induce the formation of an infection thread. This thread is constructed by the root cells, which is known as the infection thread, and it grows through the root hair cells and penetrates the other root cells nearby. The bacteria then multiply within the expanding network of tubes, continuing to produce NOD factors which stimulate the root cells to proliferate, eventually resulting in the formation of a root nodule. Within a week of infection, small nodules are usually visible. Each root nodule is packed with thousands of living rhizobium bacteria, which are known as bacteroids. Portions of the plant cell membrane surrounds the bacteroids, which are now known as symbiosomes and carry out nitrogen fixation in presence of leg hemoglobin. Nitrogen deficiency leads to development of specific deficiency symptoms. Chlorosis, which involves the yellowing of leaves, which is prominently found in the older leaves, that is the leaves near the base of the plants. Reduced branching in nitrogen deficient plants results in short and spindly plants. The slow nitrogen deficiency leads to the plants with slender and woody stems. Certain plants like tomato may develop purple-hued leaves, petioles and stems due to accumulation of the anthocyanin. Phosphorus is one of the 17 essential elements required for plant growth and development. Its concentration in plant ranges from 0.05 to 0.5% of the dry weight. It is also a key component of molecules such as nucleic acids, phospholipids and ATP. After nitrogen, phosphorus is the second most frequently limiting macronutrient for plant growth. Phosphorus can be found on earth and water, soil and sediments. It is not found in the gaseous state. Phosphorus is mainly cycled through water, soil and sediments. It moves slowly from deposits on land and in sediments to living organisms and then more slowly back into the soil and water sediments. The phosphorus cycle is the slowest biogeochemical cycle. Phosphorus is most commonly found in rock formations and ocean sediments as phosphate salts. Phosphorus is most commonly found in rock formations and ocean sediments as phosphate salts. Phosphate salts that are released from rocks through weathering usually dissolve in soil water and are later absorbed by the plants. Since the quantity of phosphorus in soil is generally small, it is the limiting factor for plant growth. Phosphorus cycles through plant and animals. When animals and plants die, phosphates will return to the soils or oceans during decay. After that, phosphorus will end up in sediments or rock formations again, remaining there waiting for the weathering cycle to process again. Soil phosphorus is found in different pools such as organic and mineral phosphorus. 20 to 80% of phosphorus in soils is found in the organic form, of which phytic acid, that is inositol hexaphosphate, is a major component. The remainder is found in the inorganic fractions containing around 170 mineral forms of phosphorus. Phosphorus also exists in the organic PI and phytates form. The solubility of these inaccessible phosphorus form is largely dependent on the pH of the rhizosphere, which changes locally and is also dependent on the microbial community in the root environment. In soil, phosphorus is usually concentrated near the soil surface. There are three major phosphorus pools which are known as the readily available phosphorus in the soil solution, the pool of P that is rapidly released from the soil to replenish the soil solution, that is labile pool, and the relatively slowly available phosphorus, which is known as a non-labile pool. Phosphorus must be dissolved in the soil solution to be taken up by plant roots. The dissolved forms of plant available phosphorus in the soil solution are called orthophosphates. Plants obtain their phosphorus from the soil solution in the form of H2PO4- and HPO42-, although the uptake of HPO42- is slower than the uptake of H2PO4-. Root hairs are the primary site for phosphorus acquisition, and in response to Pi scarcity, both the density and length of root hairs increase to explore larger volume of soil. Plants have specialized transporters at the root-soil interface for the extraction of Pi from solutions of micromolar concentrations, as well as other mechanisms for transporting Pi across membranes between intracellular compartments, where the concentration of Pi may be 1000-fold higher than in the external solution. An efflux system is also present, which plays a role in the redistribution of this phosphorus resource. The existence of high and low affinity phosphorus transport systems have been reported in plants. There are 4 families of phosphorus transporters in Arabidopsis, namely PHT1, PHT2, PHT3 and PHT4. The PHT1 family comprises high affinity phosphate H+ importers, which are mostly expressed in the roots. PHT1 is responsible for phosphorus uptake from the soil and transport to the shoot and the mycorrhizal symbiotic interface. Arbuscular mycorrhizal pathways play a major role in phosphorus uptake. The plants provide all the organic carbon requirements of the mycorrhizal fungi, whereas the phosphate is provided by the mycorrhiza. The 2 pathways by which arbuscular mycorrhiza plants absorb phosphorus involve different cell types, different Pi transporters, and phosphorus access from different regions and volumes of soil. Expression of genes encoding high affinity Pi transporters is maximum in the root apex and root hairs and is reduced in the mature regions of the root. The fungi have 2 uptake systems for phosphorus. One, a high affinity system that works against an electrochemical potential gradient, which takes up Pi from the soil via proton co-transport, and the other, a low affinity system which facilitates the diffusion of Pi across the fungal plasma membrane. Arbuscular mycorrhiza fungi express high affinity P transporters and are involved in phosphorus uptake from the soil. The expression of these transporters is regulated in response to the externally available phosphorus concentration and also on the phosphorus demand of the fungus. Solubilization of insoluble phosphorus by microorganisms was first reported by Pikoviskaya in the year 1948. Recent developments in microbe-mediated plant growth involves phosphorus acquisition through these microbes, which has led to identification of various beneficial bacteria like Pseudomonas, Bacillus, fungi like Aspergillus and Penicillium, which are capable of solubilizing phosphates. The main phosphorus solubilization mechanisms which has been employed by soil microorganisms include release of complex or mineral dissolving compounds like organic acid anions, siderophores, protons, hydroxyl ions. Second is the liberation of extracellular enzymes which are involved in the biochemical phosphorus mineralization. The release of phosphorus during substrate degradation, which is the biological phosphorus mineralization. Plants growing in phosphorus deficient conditions show retarded growth. The formation of fruits and seeds is especially reduced in plants. This not only lowers the yield but also leads to poor quality fruit and seeds which are obtained from the phosphorus deficient crops. The symptoms of phosphorus deficiency appear first on the older leaves of the plant, as phosphorus can move within the plant and is continuously redistributed towards the growing tip. Older leaves are often darkish green in color, which later turn chlorotic, that is yellow. The stems of many annual plant species suffering from phosphorus deficiency are characterized by a reddish coloration, which appears due to the enhanced formation of anthocyanins. This red coloration caused by anthocyanin has also found in the leaves of cereal crops deficient in phosphorus. To summarize, nitrogen and phosphorus are crucial for plant growth and development. Their mode of uptake and transport have been well illustrated in various plants, and their deficiencies can lead to reduced agriculture production and yields. Thank you.